In this video, I will explain the structures found in prokaryotic cells and how they divide. All prokaryotic cells have the following features. They all have a cell wall, which is made of peptidoglycan. Remember, if you're asked to draw a prokaryote, this should be drawn using a thicker line. All prokaryotes also have a plasma membrane, which has a very similar structure to that found in a eukaryote and should just be drawn with a thin line as usual. They will also all have cytoplasm, and they have 70 S ribosomes. These are the smaller ribosomes compared to the 80 S ribosomes found in eukaryotes. They also have this DNA, which is usually labelled as circular DNA, or can sometimes be called a chromosome. In addition, some prokaryotes have a flagellum for locomotion. When you draw the flagellum, you need to make sure that it is embedded in the cell wall. Others may have pili, which must be drawn as shorter than the flagellum, and those are used for attachment to other cells, and they're also involved in sexual reproduction. They can also have plasmids, just small rings of DNA, which are obviously used in genetic engineering as well. And finally, the last thing that some prokaryotes have is a capsule around the cell wall. It's sometimes called a slime capsule. Prokaryotes divide by binary fission, which is a form of asexual reproduction to produce two new cells. Multiple fission would be if you produced many new cells. Now the process is that the DNA here is going to replicate, so you have two copies of the original DNA. The DNA then starts to move to either end of the cell, and then what you start to get is a longer cell, so the cell gets longer, and then you start to see this constriction at the equatorial plate, which separates the plasma membranes, so each new cell has exactly the same genetic information.